So, since this is the simplest structure, so it is an optically inactive substance. Why optically inactive? You know, around the alpha carbon, this first carbon, there must be 4 different groups. Then only it will be optically active. Since we are having, instead of R here, if it would have been a separate group, then it would have been optically active. Since both are hydrogen, all 4s are not different. That's why it is an optically inactive substance. And this is the simplest amino acid having the smallest structure. So, in the 3-dimensional structure of protein, all the small places which are available in that compact structure, glycines will be present, because glycine will have only hydrogen and only one small group that will fit into that space. That you must remember. Glycine, although very small, its synthesis is a little bit easier, but it produces many specialized products, and it participates in several reactions. By this time, you have known a little bit about glycine. I will cover something more, and as you proceeds towards the further classes, you will know many other things. So, with that thing, I just for first slide, change it. So, this is metabolism, means basically catabolism and anabolism both. Metabolism does not include only one thing. So, we will cover the chemistry of glycine very briefly, overview of glycine metabolism, and synthesis of glycine, degradation of glycine, and specialized products from glycine, and disorders of glycine metabolism. And this is very simple. These 2 disorders are there, but they are exceedingly rare. Only for examination purposes you have to remember. But all other things are required. So, as I told you, it is a simple amino acid, means simplest amino acid, and it is also non-essential because it can be synthesized from other amino acids. Optically inactive, I clarified why it is optically inactive. And you know about the amino acid, I told you some amino acids are glucogenic, some ketogenic. So, this belongs to the category of glucogenic because it produces glucose from pyruvate. It gets converted into pyruvate and then it produces glucose. So, it is a glucogenic amino acid, and one carbon is actually given, and you will be told about one carbon metabolism. Write down one carbon metabolism is a very good short note. It can be asked, it can come as a short note, and you may be asked also in the Viva about one carbon metabolism. Glycine contributes one carbon metabolism through N5N10 tetrahydrofolic acid or tetrahydrofolate. Glycine contributes, is involved in the one carbon metabolism, and this N5N10 methylene tetrahydrofolate. There will be several forms: formyl tetrahydrofolate, methyl tetrahydrofolate, but this is related to methylene N5N10 methylene tetrahydrofolate. Okay, I will show you the slide. And I told you about in protein class, if you have not read it, I cannot do anything. In the collagen, categorically, I told you there is a special motif of tripeptide, where glycine is a third residue. Collagen needs a very compact structure, and glycine, as I told you, is a very small structure. It fits into the smallest possible space because side chains are minimum, only there are 2 hydrogen atoms are there. So, in the interior surface of the protein, that is collagen, every third amino acid is a glycine. I will show you once more. And specialized product produced from glycine, heme synthesis you have been told, heme class has been covered, taken. Heme synthesis? No. Okay. So there will be a separate class on heme. If during that time, meanwhile you write down, I mean, what are the things that are produced from creatine? One chart I will tell you, this chart will be available in the book. This is the heart. At least you have to remember everything of this chart only. And second part is glycine cleavage system. These are the two things you have to remember in glycine. So why I am telling you to note down, so that while you are writing, you will start memorizing also. See, glycine, where from it will be obtained? At the breakdown from protein. If the digestion of the protein in the intestinal tract, it can be obtained, or from intracellular digestion in the body, from the endogenous protein breakdown. So it can be obtained from protein, one source. Second source is from threonine or serine, and third one is glyoxylate. Write down. Glyoxylate is another source. And since, see here, ammonia is there, carbon then the glycine can be produced. So that N5N10 tetrahydrofolate contributes this, you see, carbon, ammonia, carbon dioxide, ammonia. Carbon dioxide will be available here. This group is there, so carbon dioxide we can get from here, and ammonia we will get here. Only thing is that we need one more carbon. That one more carbon is given by N5N10 tetrahydrofolate. That's why I told you it is involved in one carbon metabolism. So only one carbon will be donated by that, and it will glycine will be formed. So there are the 5 sources of glycine. One is protein, digestion protein, either extracellularly, I mean in the digestive tract or from intracellular, serine, threonine, glyoxylate, and from carbon dioxide, ammonia, and one carbon from tetrahydrofolate. Now the glycine cleavage system, now if you break down glycine, this part is available in the book, you write down and memorize, so that at least you will be able to answer this question. That's my point. So glycine cleavage system produces ammonia again. If you break down glycine, so then ammonia will be produced, one carbon will be produced again. If you break down, carbon dioxide will also be produced, isn't it? Since these can be synthesized by this thing, if you break down, you will get the same product. Serine, as I told you, I am showing you in the next slide. From serine to pyruvate to glucose, it is a glucogenic amino acid, I told you. So from glycine, glucose can be obtained, but through serine and pyruvate, not directly. So it is a glucogenic amino acid. It can be converted into cysteine, it can be converted into choline. This glyoxylate, you have seen that it can be formed from glyoxylate, but again glycine can form glyoxylate also. It's a reversible reaction. Serine, threonine, serine to glycine is a reversible reaction. Glycine, glyoxylate to glycine is also a reversible reaction. So I will show you all these things in detail. Now another thing is the creatine. Creatine is produced from glycine. I will show you how it is formed. And from creatinine is produced, and that creatinine is excreted in the urine. Okay. And another important, this C4, 5 and N7. You see the purine ring, if you remember, Purine ring is just like that. You remember this one is the purine structure, and purine structure is just like this. So 4, 5 and 7, that N, that purine is contributed to the 4, 5 position mein, this carbon is there, and 7 position is nitrogen is there, and that is contributed by glycine. Heme synthesis occurs through ALA, delta amino levulinic acid, and glutathione has been repeatedly told that it's a tripeptide made up of 3 things that you must remember, and that maintains the redox potential of the cell and RBC membrane integrity. Bile salt. Bile acids are produced from cholesterol. Bile acids are cholic acids and chenodeoxycholic acid. Now, when they produce salt as sodium glycocholate or sodium taurocholate or sodium chenodeoxycholate, then they are called bile acids, bile salts. So, bile salts will be told you in detail during bile salt practical exhibition. And then conjugation, it conjugates with many things. One is hippuric acid. This test used to be done where we are students, but this test is no longer used as a liver function test, because it indicates the liver's capacity to conjugate. So, it used to be, it is basically a historical test. So, this is the thing you have to remember. Once you remember this slide, then rest of the things you will be able to write down, because by this time if you will read many things, many reactions, and only thing of remembering the points, then you will be able to write it down. Now, I am showing it. Synthesis from 4 sources, as I told you, basically 5 sources. One is protein breakdown, that I am not covering, because by default it will be produced. Second is serine. From serine, you see the carbon which is attached to carboxyl group is called alpha carbon. The carbon which is attached to carboxyl group is called alpha carbon. Okay. Now, any structure which is attached either above, it is a first position is alpha position. Now, if anything is attached to this position, suppose CH2 or CH3, so this is a beta position. Okay. Now, if you look at the structure of serine, this structure is the first part. You see, look at this structure. You look at this structure. It is having a structure like this. This structure, you remember, look at this. Serine is here. Serine is here. This structure is having a alpha carbon here, and this is beta carbon. Now, if you cleave it, then you will get glycine. So, this is a reversible reaction. Note down, only thing is that the enzyme name, serine hydroxymethyl transferase. You see, again, methylene tetrahydrofolate is involved, 1 carbon metabolism. So, only thing is that it is breaking the serine part, and it is breaking into 2 components. So, basically serine to glycine. See here, the N5, N10 methylene tetrahydrofolate is required. So, threonine aldolase, threonine. You look at the structure of threonine. You see, this is the structure. This structure is basically glycine. And if you remove one H from here, then it will make a glycine. Now, only thing is that CH3 and CHO will be available. So, basically that will be forming acetaldehyde. Do you get my point? There is a simple thing, only thing I have to remember, the name of the enzyme. What type of enzyme it is? In which class? Which class of the enzyme? From the reaction you tell. You try to, whenever you are reading about enzyme, you try to remember, then you will remember the classification immediately. All the classification will be in your mind. Okay? Yeah. There is no water requirement here. Okay? This is actually learning biochemistry, technically. Second, glycine from glyoxylate. Glyoxylate, you see, it is a keto acid. So we need an amino acid. So amino acid can be added to a keto acid by transamination reaction. It is as simple as that. I have told you during transamination that when an alpha keto amino acid is converted into through a transamination reaction, that keto acid gets converted into an alpha amino acid. And the amino acid which is contributing that amino group itself will get converted into keto acid. So ping pong mechanism I told you. So where in a transamination reaction, one is an alpha keto acid, one is an alpha amino acid. One is donor, one is acceptor. The keto acid which is taking amino group from the alpha amino acid will be converted into an amino acid. But that which is donating amino group will itself be converted into keto acid. That is what you can see here. So here keto acid is there, alanine or glutamate can act as a amino donor, amino group donor. So it will get converted into pyruvic acid or alpha ketogenic acid, and instead glycine will be formed. As I told you, it is a reversible reaction. So again, if you add glycine, it will form the glyoxylate. So you see here, this is a reversible reaction. So once glyoxylate is there, you add glutamic acid, and transamination reaction always require pyridoxal phosphate. So that glutamic acid is getting converted into keto acid. Glyoxylate is getting converted into an amino acid as glycine. Okay. So glycine synthesis, again you see, glycine can be synthesized. I told you in the beginning that it requires three things. One is carbon dioxide, one is ammonia, and one extra carbon. That extra carbon is provided by tetrahydrofolate. Methylene this tetrahydrofolate, and this will be converted into glycine. Now reverse reaction is also possible. So glycine breaks down into carbon dioxide, ammonia, and ammonia carbon dioxide can combine with glycine. So this is a glycine cleavage system. It's a reversal. Means instead of when this reaction proceeds in this direction, that is called a glycine cleavage system. When it is reversed, then only glycine will be produced. I will show you the glycine cleavage system in the next slide. Now, glycine cleavage system, only this part, the two charts you have to remember, only this one you have to remember, and you have to remember this as a short note. You concentrate on this one. This is glycine cleavage system. You try to understand this thing. So, glycine cleavage system contains three enzymes, note down. One is glycine dehydrogenase, which is also told as glycine decarboxylase. Basically, glycine dehydrogenase is decarboxylating enzyme. You see, this is number one is decarboxylating enzyme. So, during the reaction, carbon dioxide is coming out. Second is an amino forming amino methyl transferase. This one. You see here, tetrahydrofolate is there. It is getting converted into 5,10 methylene tetrahydrofolate, and ammonia is produced. And C3 is dihydrolopamide dehydrogenase, because this is required to oxidize NAD to NADH. Okay, this you will see in the next slide. You try to remember this thing. This part you have, this is a complex thing. This H protein is attached to it and 3 more enzymes are attached to that structure. You see here these are called P, N and T, P, T, L. They are basically representing the same thing as I mentioned here. These 3 enzymes you have to remember at any cost. This is the only part a little bit difficult, but you have to memorize. That is the only difficult part of glycine. So, you see there are four proteins are there: P protein, T protein, L protein and H protein. I am showing it here. You see H protein is here, P is there, T is here and L is there. Okay. So, these 1, 2, 3 are basically P, L and T. So, glycine undergoes oxidative deamination. As I told you that when you do an deamination, when it is coupled with oxidation, that is called oxidative deamination. Then it produces ammonia and carbon dioxide and 1 carbon unit of methylene, tetrahydrofolate, that I told you. And this is the major catabolic product. What is the major catabolic product of glycine? If you say that, you see the completely breakdown. So, 3 structures will be produced only. So, this 3 structure is the major catabolic product, and the glycine complex cleavage system is a multi-system complex. As I told you, H protein is there, it is in the central part, and in the periphery there are 3 proteins which are basically enzymes. Now, you see, look at this structure. This lopamide dehydrogenase is required to oxidize this NADH. See, lopamide is there, lipoamide. This is reduced form. This is getting lipoamide. This NAD is getting converted into NADH. For any cycle to operate, you require a constant supply of all the substrates. Now, if you want to break this glycine into these 3 structures, amino acid, carbon dioxide and this one methylene tetrahydrofolate, you see this is a breakdown product. This is a cleavage system, clear cleavage system. We are cleaving glycine into 3 components. One is carbon dioxide, one is ammonia and one is 1 carbon unit, that is methylene tetrahydrofolate. So, this is a complete breakdown. If you reverse this thing, then from all these 3 structures, glycine will be produced. So, when the body is in need of glycine, it will be synthesized in that backward direction. When the body wants to break it down, it will break it down in that way. That is the thing. Okay. Now, glucogenic pathway, as I told you, that it does not contribute directly to glucose, but through serine. So, glycine is converted to serine, as I told you. The serine conversion is a serine methyl transferase, is a reversible reaction. So, hydroxymethyl transferase reaction, glycine is converted, and then serine to pyruvate by serine dehydrogenase to an amino acid, and then it undergoes further conversion to pyruvate. So, this pyruvate acts as a gluconeogenic precursor. If you recall gluconeogenic precursor, pyruvate is there. So, that is the reason why it is called a glucogenic amino acid, and this is the reaction of serine to pyruvate. Only here the name of the enzymes will be required, and this will be taught in other classes also. Only thing that chart you remember, only the name of the enzymes you have to write down. What are the specialized products that are produced from glycine? This was there in the chart again. I am telling you, first is conversion to serine. That in the first reaction I have told you. Second is a glucogenic amino acid, I have discussed. Third one is glutathione. This production I will tell you. Heme production, purine nucleotide, and it acts as an inhibitor neurotransmitter also. Neurotransmitter, as an acts as a neurotransmitter, you can write. Inhibitor transmitter not always, but it can cause excitation also. So, normally it acts as an inhibitory transmitter, but when in an excess amount, then it causes an excitatory inhibitor also. So you can write down only neurotransmitter and conjugation with bile acids. It conjugates with bile acids. I will tell you all reactions one after another. Creatine, creatine phosphate, and creatinine. Creatine and creatinine are not same thing. Creatine phosphate is there, it is in the muscle. Creatinine is produced from creatine, and that creatine is excreted through the urine. So high level of creatinine indicates a poor renal dysfunction because kidney is unable to excrete creatinine. This will be dealt in detail in your practical class as well as during renal function test. Then you will understand. For the time being, you try to know only that creatine phosphate and creatinine is produced from glycine, because the creatine is the precursor of creatinine. Okay. So contributor of one carbon pool, as I told you, this is one important part, and you read that part. Somehow it is difficult to remember, but just memorize that cycle, one carbon. Even today itself, you find out where is one carbon metabolism is written. That small part is written. It will take about maximum half an hour time, but there is a high probability that this question will come, and it came during my MD examination also. It appeared. So it is so important. Usually there is a mistake. I mean, we cannot remember, but it is so important that even during MD examination it was given. So one carbon metabolism is a very important issue. Another is detoxification of benzoate to hypurate. As I told you, this is a historical significance. So these are the metabolic functions, and there is metabolic functions of glycines also. So creatine, creatine phosphate, and creatinine. Here you note down here. The first reaction, glycine is involved here. 3 amino acids are there. One is glycine, one is arginine, and third one is methionine. Methionine doesn't participate always in reaction, but the active form of methionine, write down, active form of methionine is S-adenosyl methionine. Active form of methionine is S-adenosyl methionine. The first reaction, this reaction glycine to arginine, which forms guanidoacetic acid, you see here this guanidino group. Yesterday during urea cycle, I told you that this structure, this particular structure was there in the arginine. By the arginase enzyme, this structure was removed and it was produced an urea. So this is a guanidino group. That guanidino group is transferred to this structure, and guanidoacetic acid is formed. Okay. So ornithine is again coming, you see, again ornithine is coming here. Yesterday I told you about ornithine during urea cycle, ornithine cycle it is also called. So this guanidino group is transferred from this arginine to glycine, it is forming guanidoacetic acid. This is occurring in the mitochondria of kidney and pancreas, then it is released in the circulation. Okay. The second reaction, S-adenosyl methionine, is the active form of methionine. It donates the methyl group to guanidoacetate to form creatine, and this methylation reaction occurs in the liver. So one was there in the pancreas, you see this reaction, first reaction was there in the kidney and pancreas. First reaction was there in the kidney and pancreas, and second reaction is occurring in the liver. So liver is the production site of creatine. Okay, this methylation will take place, guanido, glycine, arginine, amido transferase. This group is again transferred to guanidoacetic acid, and creatinine is finally produced. So S-adenosyl methionine is step 2. Step 2, S-adenosyl methionine group is being converted. So one methyl group is being added to it. So this is basically called the structure of creatine. Creatine, remember. Now we got creatine in 2 reactions. The third reaction, now creatine is getting converted, it is getting phosphorylated. by the enzyme creatine phosphokinase. And this is again the reversible reaction. And here you see ATP is converted into ADP, and one high energy phosphate bond is there stored in the creatine phosphate. And this creatine phosphate is present in the muscle and acts as a ready source of energy during muscle contraction. When somebody needs to perform a sudden reaction, a sudden, muscular contraction, maybe running, jumping. So, immediate energy is obtained from creatine phosphate, and this creatine phosphate, this high energy bond is stored in the creatine, and from that creatine, once the creatine phosphate is released, it is back into creatine. So, creatine phosphate acts as a immediate supply source of energy for the muscle. Now, creatine gets converted into creatinine through a spontaneous cyclization of creatine. Okay, spontaneous cyclization, non-enzymatic reaction basically. So, there is a cyclization of this is creatinine, and this creatinine is excreted. Now, out of for your own information, in your OPD or IPD, there will be several questions, and in the era of Google, everybody is a doctor. They will ask some questions. It was a story of 2000, when I had my junior, senior to me by 15 years by position, he was, my junior, he was having an MSc and PhD in organic chemistry. One patient asked, Dr. Babu, what is creatinine? So he tried to explain creatinine. He failed miserably. Then I was in my room, and he brought the patient. Doctor, please address to the patient. He asked me what is creatinine and what reply I have given to him. I knew that he has tried his level best to make him understand what is creatinine. Is it possible for a lay person to understand what is creatinine? Is it at all possible? So this type of question, please tell it, what has happened, what is this thing? The moment the patient entered into my room, what is your question? What creatine? Well, yes, it is a spontaneous cyclization of creatine which is produced from guanidinic acetic acid. Patient simply put down, left this place. You have to say something, and something correct. Only hearing this thing, the director of Bhilai told me, next time somebody comes to me for irritation, I will make an emergency call to you. So, what is creatine? Creatine is basically cyclization form of creatine which is produced from guanidinic acetic acid. I have not said anything wrong. But that silenced the patient and he left. So, at times you have to be very clear who you are dealing. So, there will be many situations. This is out of the track. During muscle contraction, as I told you, since this creatine phosphate is produced, this first reaction you see, creatine from ATP to ADP is there. So, that means one phosphate is getting released. This phosphate is stored here. So, that acts as an immediate source of energy, as I told you, and during muscle contraction, energy from hydrolysis of ATP is obtained, and ATP is regenerated by hydrolysis of creatine phosphate. This is called Lohmann's reaction. Again, if you require, it can be converted back also, creatine to creatine kinase. So, this energy which was stored there now is getting released. Here you see, it is getting released. So, creatine phosphate is a stored energy in the muscle and acts as a ready source. So, to summarize, creatinine, it is produced from glycine and arginine in the first step. It is produced in the kidney and pancreas. In the second stage, it is made in the liver, and then it is transported to muscle where it is stored in the form of creatine phosphate. The reaction is a spontaneous one, and this creatine formation is a spontaneous one, and but creatine phosphate is present in the muscle. Okay, creatine phosphate is present in the muscle. Now, one important part is here, since the body muscle mass is usually fixed. So, in a given individual, creatinine level in blood remains almost stable. It is a very useful parameter. Now, if the kidney function remains alright, the creatinine level will be normal. Now, it is excreted through the urine. So, if the kidney function is deteriorated, that means it will not be able to clear the creatinine. So, creatinine level will rise. And the patient, what I referred to you, was basically a renal failure patient whose creatinine level was high. And since it was high, it was his concern what it is. Because his creatinine level is high, where from it is coming, he wants to control all those possible sources, but he cannot control the kidney disease which has happened. So, special metabolic functions I covered all something there. This part I have creatine I have covered. Purine nucleotide I will tell you, glutathione I will tell you, and conjugation with bile acids again I will tell you. So, synthesis of heme, just note down one point at least, starting sentence you can write. Glycine condenses with succinyl-CoA. This is the first step of heme synthesis. ALA synthesis is there. So, ALA synthesis is required. So, glycine is a precursor of heme synthesis. So, this is one function, synthesis of heme. That structure, if you remember, only you have to write down the reaction. On the against this is the name, you just write down heme synthesis, and you write down this reaction. But that will cover the functions of glycine, because everything you cannot write in the examination hall because of time constraint. Okay, so everything you will get it in the book. Second, this one you will note down. You see synthesis of purine ring. So, purine ring, as I told you here, this 4, 5, there is a carbon ring, and 7 position is nitrogen. So, this 7th nitrogen, the glycine is having 3 things. One COH, 2 carbon is here. So, that 2 carbon is coming, contributing 4 and 5, and 7th nitrogen is there. 7th nitrogen is coming from ammonia. So, C4, 5 and N7 is contributed by glycine. While you are writing, you try to memorize, so that once you read the book, today it will be complete. Synthesis of glutathione. Glutathione contains 3 amino acids. One is glycine, is an important amino acid. So, there are 2 forms of glutathione. One is reduced form, GSH, and oxidized form, and there is continuous changeover from oxidized form to reduced form, and body tries to keep glutathione in reduced form, so that its RBC membrane integrity is maintained, it does not get lysis, and also redox potential of the cell. So, if by chance instead of GSH, too much of GSH is formed, that structure cannot be maintained, and that happens, then RBC membrane will be, structure will be lost, and there will be hemolysis, RBC will be ruptured, and that happens, you will read it. I think this has been told also during your HMP shunt. So, this structure in the initial stage, glutamate and cysteine is formed. So, glutamyl cysteine is formed, to which glycine is added, and then this glutathione synthesis occurs. So, basically what I want to emphasize, glycine is one component of glutathione. Conjugation reaction. So, the bile acids are produced from cholesterol. Cholesterol, everybody is telling cholesterol, low cholesterol diet, cholesterol, it's a bad cholesterol, good cholesterol, this, that. Cholesterol is causing heart disease, but cholesterol performs several important functions. I am really in argument with the clinicians. that why you try to decrease the cholesterol, because cholesterol is present in all cell membranes. Cholesterol is present in this fat is present in the neural tissue, and you are having only one organ, that is heart, and to protect the heart you want to destroy the whole body. All cellular membrane structure contains cholesterol. Steroid hormones are synthesized from cholesterol. Vitamin D is synthesized from cholesterol. Bile acid is synthesized from cholesterol. Now, if you remove cholesterol, decrease the cholesterol, all the functions will be compromised. And that's why those who are taking a cholesterol lowering agent too much, they don't feel well, because somehow what I personally believe, that lowering too much of cholesterol in the body will cause some amount of cellular alteration. In fact, when I took cholesterol lowering agent at a little bit higher dose, believe me, I had so much of muscle pain that I had to sell my Maruti 800 car because I could not get up from the seat. I had to purchase a Wagon-R car, but the doctor don't want to believe. Everybody's system is not same. Once I stopped it, everything was normal. From Maruti seat, 800 seat, I could not get up. So it was a very serious condition. It's only with 10 milligram only. It happens from person to person. So bile salt, see cholic acid and chenodeoxycholic acid are produced from cholesterol, and they are conjugated with glycine to form glycocholic acid and chenodeoxycholic acid. Okay. So when they are acid, when they combine with the sodium salt, they call sodium glycocholate or sodium taurocolate or sodium chenodeoxycholate. These are all basically bile acids, bile salt. The bile salt examination you will tell, you will be taught during a practical examination. At the time, try to remember the glycocholic acid can be conjugated with cholic acid can be conjugated with cholic acid. Cholic acid can be conjugated with glycine will be conjugated with cholic acid, and glycine will be conjugated with chenodeoxycholic acid also. It is a constituent of protein. It's a small amino acid, so it is there, although it is non-essential amino acid, but it is present in protein. And since this is a very small protein, as I told you during protein class, that there are small bends. At that bends or loops, at that loops the glycine is present. This is a very small structure. And the proline structure I have told you, every third amino acid is a glycine. Glycine XY, glycine XY, glycine XY. This is a structural motif of collagen. So that collagen becomes a very compact structure. So every third amino acid in collagen is a glycine. So it acts as a neurotransmitter, not always inhibitory. As you can see, brainstem and spinal cord, it basically opens the specific chloride channels. But at moderate level, it disrupts or inhibits neuronal traffic, inhibitory role. But at a very high level, it causes excitation. So it is, you write down, it is a neurotransmitter. It acts as an inhibitory part when it is in a moderate concentration. It will act as an excitatory neurotransmitter when in high concentration. In laboratory, in your clinical practice, you will use glycine. Old-aged persons undergo prostate operation. Now, after prostate operation, urinary bladder is washed, and glycine, 1.5% glycine is used in that buffer as an irrigation solution of the urinary bladder. So, urinary bladder is irrigated with a solution after prostate operation that contains glycine. And in laboratory, we use Trees glycine buffer or glycine buffer. So, these buffer we used to prepare during our time, now these are all commercially available. So, during electrophoresis we need this buffer, even as a wash buffer we need glycine, and also western blotting, which will be told during your molecular biology class how western blotting is done. So, there is a washing step, that washing step requires Glycine particularly, remember, at least electrophoresis. So you see tris glycine buffer is there, means not only glycine but tris is there, SDC is also there. So earlier in our time we used to prepare it using our own hands in a laboratory, in a container. Benzoic acid is used as a preservative. Benzoyl acid combines with glycine to form hippuric acid. It used to be a test of liver function test of the conjugation capability. Now it is no longer used as a test, it is a historical test. You just write down benzoic hippuric acid test is a historical test. It is not done nowadays. At least I have not seen. So metabolic disorder of glycine only 2. What is non-ketotic hyperglycinemia? If there is a defect in the glycine cleavage system, so glycine will not be able to break down. So then there will be hyperglycinemia. There is one form of hyperglycinemia which is associated with ketosis. So that's why its name is non-ketotic. That is a separate disorder. Now when you get an hyperglycemia, hey, leg pen. When glycine is excreted in high amount, produced in high amount, that is called hyperglycemia. Now in that condition, if you get only glycine but no ketone bodies in the blood, that means it is a non-ketotic hyperglycemia. So basic problem is in the glycine cleavage system. If it is associated with ketone body also, then it is a ketotic. So that is a separate disorder. It is not glycine cleavage system. So there is no effective metabolism management. That will be true for many diseases. Glycinuria is a rare disorder in the kidney. From the proximal columnar tubule, amino acids are absorbed, and glycine is also a part. So glycine cannot be absorbed by the renal tubules. So glycine will be excreted in the urine. So that will cause glycinuria. So this is due to defective renal absorption, and it can form oxalate stone. So glycine, from glycine to glyoxylate, glyoxylic acid to oxalic acid. So leafy vegetables, vitamin C and ascorbic acid, from there oxalic acid is produced. So those persons who are having this problem, glycinuria, we have to advise not to take these items so that they can produce the formation of oxalic acid. That is the mechanism of oxalic acid formation in glycinuria. Glycine gets converted to glyoxylic acid, from glyoxylic acid, oxalic acid is formed. So basically oxalic acid stone will get, or oxalate stone will get. Because as I told you, it does not remain in the acid form, always in a salt form. So oxalate stone will be there. So primary hyperoxaluria is the protein targeting defect. The enzymes in the subcellular organelles, enzymes are compartmentalized. Some organisms are very much specific in our organ, some organisms are common, and some enzymes are present in not organisms, as I told you wrongly. Enzymes are present in specifically in some component like lysosome. It contains hydrolysis. Now peroxisome, it contains oxidizing agents. Mitochondria, we are having TCA cycle enzymes, electron transport chain enzymes. These are all specific. But some enzymes are common. As I told you yesterday, that there is a counterpart of TCA cycle also, malate, fumarate to malate, to oxaloacetate. There is a counterpart, so these are also present. Now, by chance, after what happens after protein synthesis, the proteins are sorted out. Suppose in a post office, many things are there. Many things have come, and you are now sorting it out. Now, if you are wrong, this item is supposed to be sent to, say, Santirikata Medical College. Instead of you have sent it to Biswavarthi. What will happen? It is just like that. It is a protein targeting defect. But some enzyme, this enzyme particularly, a glyoxylate amino transferase, it is normally present in peroxisome. Now, because of the faulty mechanism, it is transferred into mitochondria. This will not be able to perform its own function in the peroxisome. So it will not be able to perform its function in mitochondria as well. So, that function will be compromised, and that will lead to formation of glyoxylate or oxalic acid. So, each protein when it is formed, it is targeted. This is called protein targeting. You will be told in your class also after protein synthesis that specifically proteins after synthesis, they are targeted, and that there is a target sequence also, so that the proteins are sent to the place where it is supposed to be present. If by chance there is a faulty procedure, maybe due to a defect, then proteins, this is a classical example of protein targeting defect, where this glyoxylate amino transferred, instead of sending it to peroxisome, it is found in, it is sent in mitochondria. So, it cannot perform its own function, and of course, it causes oxalate stone, as I told you. And nephrolithiasis is basically, the term is basically called stone in the kidney. So, when there will be a stone in the kidney, that stone, if it is a small one, it will try to come out through the ureter, and when it will come out through the ureter, there will be a severe pain, because this is a small structure, and when there will be, it will try to pass, there will be bleeding. So, that blood will come in the urine. So, that is what is called hematuria, and this is this pain from loin to groin, because it will come from kidney to bladder in this side. So, there will be pain. This is called the renal pain. Typical renal pain, basically when the pain is from loin to groin, first think of renal colic, and it is most likely due to a renal stone. External oxalosis can also occur, because it will be present in high concentration. So, in other organs it can also be present. Okay. So, as I told you, this is a peroxisome. Instead of peroxisome, where it performs its own function, this function is compromised. It cannot no longer function. So, that is the reason why it happens. So, here, as I told you also earlier, that from glycine, glyoxylic acid is formed. So, that glyoxylic acid to oxalic acid. So, this is the basis why oxalate stone are present in glycemia or glycinuria. Okay. So, type 2 hyperglycemia is a milder condition, a deficient activity. This is a separate disorder. So, cytoplasmic glyoxylate reductase, these are exceedingly rare condition. Now, when in general, if there is any renal stone in any patient, the first and foremost advice is to advise him to take excess amount of water, so that excess amount of water will be passed, until unless it is contraindicated, because all patients you cannot say that you drink water as much as you can, but those who are having renal stone, if otherwise healthy, water is not contraindicated excess amount, ask them to take adequate amount of water, so that they will get little chance to form the stone to occur. Those who do not pass urine, try to hold out the urine, there is a high chance that the concentrated urine form renal stone. So, it is mandatory that you pass at least urine 5 to 6 times per day, and during summer also try to pass urine at least some more time if possible. You drink as much as water, because you don't know which disease you are suffering from, or whether you are prone to stone or not, but once you suffer from a renal colic, you will realize what pain you are suffering from. So, then minimize dietary intake of oxalate. Oxalate content, if it is an oxalate stone. If it is other stone, then there is no need, and it is present in the leafy vegetables, as I told you. So, this is the end of today's class. I think not much to remember, except that first chart what I have shown, that you remember and try to note down, so that in one page that chart will be written along with the reactions, and the second question will be glycine cleavage system. So, then the glycine part will be covered completely.